Thank you so much for inviting me for this talk. I feel so honored and privileged actually. So thank you, Dr. Jagannathan and Dr. Vitha and Dr. Pantali, right sitting there. So I'm also looking forward to collaborating with SVT, and I'm very excited about what is to come. So today, first I want to give a disclaimer. I'm not a food security expert. I'm a precision nutrition. I do work in this specialty. But this seems to be a very interesting area. And when Dr. Madan suggested to me, I started looking into it. And it is, I'll tell you how we can link it to it. So before So I'll talk a little, you can open the hood. I'll talk a little bit about what I do, what is my research. So I have, like we said, like Dr. Rekha said, I have a background in nutrition, PhD in nutritional sciences. I've studied nutrition all my life. But when we were doing a study in, when I started my PhD first year, we were doing a study in doing a weight loss management program or weight loss program in women from low income group. I realized that there's so much variability in how people respond. We are giving the same diet, we match them for age, of course sex because they are all women, and then still we give the same type of diet, and they do say, of course, compliance is a problem, but there's so much variability. So I started learning what could be the baseline. At that time, we only knew about genetics. So I took a postdoctoral fellowship in genetic epidemiology. And then I started thinking about how we can incorporate genetics into nutrition. So that's how I landed with nutrigenetics and nutrigenomics, and now it has expanded into precision nutrition. So that's what I'm going to talk about. What is it actually, precision nutrition? So my main, I would like to study every metabolic pathway, every nutrient, but you can't go in depth. So I've been focusing on purines and one carbon metabolism and how they affect disease risk. Mainly focusing on obesity, diabetes, and neurodegenerative diseases like Parkinson's and Alzheimer's. And I do mostly human studies. I don't work with animal studies, but we do clinical trials where we are trying to give diets or drinks based on their genetic background. So we pre-screen them, those who have a certain genetic background and those who do not. And then we see how they respond to whatever food we give. And then we also do cohort studies. Cohort studies is basically recruit people irrespective of their, like if you want, I have a study going on childhood obesity. So we recruit children and their parents because children cannot be studied in isolation. So we collect all their blood samples and ethnometric data, demographic data and everything. And then we analyze to look, and we measure genotypes and biomarkers and see if there is any correlation. And then once we have this, then we also do some cell culture studies. You want to treat the cells with certain like fructose or caffeine, which are of interest to me, and see how it affects genes involved in inflammation, oxidative stress. So that's what, and eventually, of course, they are all linked to obesity, diabetes, and degenerative diseases. So today I will just mainly talk about two topics. First is what is precision nutrition, and how we can study the individual responses to diet and nutrition. And second is how can we apply it into food insecurity, because it's so important. This is a standard slide which I always use. We all unhealthy diets and undernutrition and malnutrition is a big problem. I'm not going to dwell on it because you all are doing the same work. What concerns is a change in dietary patterns. People are moving for more convenience foods, comfort foods, and also ultra-processed food. Now there's a lot of people are recognizing that it's not good, but still its consumption is very high. So according to the general recommendations or RDAs, we have any diet which is high in fiber, low in fruits and vegetables, 5 servings of fruits and vegetables, and low in salt, sodium, or red and processed meat. or even low in refined starches or grain, sugary beverages, and avoid energy dense and highly processed foods. So we are giving this recommendation to everybody, and let's assume it's a perfect world and everybody is following. Do we see the same response? Not really, actually. You will be surprised that even though everybody is following, you will not see it, because underlying metabolism is totally different from it. It's based on your genetic makeup and also your ethnic, where you're from. Here we are all Asian. It is interesting when we do studies there, we are doing Asians versus African Americans or Caucasian, but Asians are not same. We have to separate them South Asians, East Asians, and within South Asians we have so much variability. So can we bring them in, make them into groups and see how each group responds? So this is what I just talked about. So we not only are what we are based on our genetic makeup and what we are also inherited from. And actually I should have added one more, how we are exposed to the environment. What environment are we exposed to? Let's say even children of the same parents, they are exposed to different environments, they respond differently. So, yeah. So I want to just talk about a small study. It's a ZV et al. If you have time, or I can share the PDF. It's not mine. It's an old paper, but very interestingly, what they gave it was they gave different types of diets to people, 800 people, and they looked at their responses. So the first graph, you see that they looked at 1 week and so much variability. This is only glucose. Okay, glucose response. They had the continuous glucose monitoring, and then you see that. And then when they started looking deeper into 1 day, you'll see that they gave same meals every day, but there was so much variability within the person. Yeah, this one. Okay. Yeah. So you can see the variability. So this is a study which we did. We have about 60 people, we called them and did an oral fructose tolerance test. And continuous measured like for baseline 30 minutes, 60 minutes, 90, 120 minutes and 240. And you see that there were only 60 people, but everybody responded differently. And we had a very narrow age range. It was 40 to 50. Within that also, we see that men and women, even within women, you see. And I didn't include, but we had another one based on, this is serum uric acid, which is interesting. We also looked at Caucasians and at African Americans. And similarly, within African Americans, there's so much variability. So this makes us think that, okay, do we just say all people eat the same thing, or 50 to 70 age group, you are given this RDA? Is that right? But it's practical. You know, you have to imagine when they create RDAs, you can't expect them to have RDAs for each person. So what is the solution? Oh, I'm going back. Oh, yeah. Right. Yes. No, the other way is going back. Oh yeah, this is what. So this is what I want to talk. Oh, did you do anything? Okay, I thought magic. I didn't do anything. So what do we mean by precision nutrition? So sorry to contradict, but it is not an individualized response. Preciseness can be focused to individual, but it is here we are talking about specific population subgroups, responders versus non-responders, and that is what is precision nutrition. This individual will come later, because right now we are at a stage we do want to look at public health, which is the entire population. Then we go in little bit specific population subgroups. So we are looking based on their ethnicity, or males and females, or age groups, older and younger people, and also responders and non-responders. How you respond to a certain drink, and how do you not? Some people, if we see, we also measured CRP as an inflammatory marker. We saw that everybody responded the same way. So I just didn't put that graph because they are straight lines, which is so strange because African Americans and Caucasian, everybody responded the same way. So we said, okay, ethnicity is not affecting the way they respond, age is not affecting, what is affecting? we started looking into genetics, you will see totally different responses. So this is what I'm going to talk about today, little bit, where we are talking about population subgroups. We do want to go to personalized level, but I think at this stage we are at the precision in population subgroups, and these subgroups can be anything, like I said, based on sex or based on response, but this is what we are talking about. So we know that there is individual response. How do we group them? How do we make this subgroup? So if you look at the top graph, you will, when you start putting them together, you will automatically find that some people respond very well to the diet or drink or whatever, and some people do not at all. So we combine them into one, like separate responders versus responders. So this is mostly done, there's a lot of variability. CRP maybe they'll be responsive, but maybe not glucose or maybe something else. So you have to think about what is your outcome of interest. So just because they respond very well to inflammatory marker doesn't mean that they're responsive. That is why it's so difficult to find a proper individual diet plan. Otherwise, everybody would be cured and everybody would be no need for studying this. But what I'm saying is there is a lot of individual variation. So coming to it, so what is responsible for this individual variation? So the nutrigenomics is what I do extensive studies. Basically, I'll talk a little bit more about it. But other things are gut microbiota, because I'll talk about that. Then you have metabolomics, which is the metabolites, which is the final product of metabolism. What we eat and it gets metabolized and affected by genetics, epigenetics and everything, and finally what is left in the metabolite, that is your final product. And this is the phenotype, this is an approach, but basically what are your dietary habits, and also this picture I took from this paper, but they don't include exposures, other exposures, like your lifestyle or environmental pollutants, or so those are not included in this one. So I'll talk a little bit about nutrigenomics. So this is basically talking about nutrients and human genome. So first one is, some people actually use nutrigenomics for everything. Some people say nutritional genomics, they like to say. And so, and nutrigenetics is how individual genetic differences influence metabolism. Like I'm different from her or her, we all respond differently. But when we do studies, a lot of age can be responsible, or sex can be, or ethnicity. So after adjusting all for that, we look at what genetic variants are affecting that. Nutrigenomics is slightly different, is how genes are expressed in the body. So I don't know how much genetic background you have. We have a DNA sequence. It is expressed as RNA, then it gets converted to protein, and protein is the one which actually does everything, like enzymes or catalyzes reactions. So this is what we are talking about, what we are eating. We'll find that if I drink a lot of fructose drink, like your sodas and other things, some set of genes will be more expressed than others, or some RNA will be more than compared to others. But if I change it to, like, let's say milk, then other genes will be expressed. So what you are eating will affect what is expressed. So the little bit detail, those who do not have a genetics background. So our DNA sequence is made of 4 nucleotides. Very interestingly, 99% of us are same. We all ABCD and we all have same. That 1% makes all the difference, which is strange, isn't it? But that's the way it is. So you see that this is genetic variant. Everybody has, like, ancestrally, we all have A, but some people, due to some mutation, can change into G. It looks like only one nucleotide is changing. But remember, we have millions of genes and all of them have these changing patterns. And it depends upon where the SNP is, it's called SNP, single nucleotide polymorphism, and it's pronounced as SNP. So, which part of gene is it located? Is it like where exon, which is converted mRNA, or it's in tron, which is sliced out. So we don't know. So based on that, it can make a difference. The other one is your sequence may not be affected, but how the RNA is formed, that may be affected. Okay? And then how RNA is formed, and then how the protein is formed. So if we eat a different, I'm just mainly focusing on nutrition, but how RNA is expressed can be what physical activity, other lifestyle factors, physical activity, exposure to pollutants, and there are so many other things, but I'm focusing on the nutrition part. So nutrigenetics, this is a classic example of nutrigenetics, and which I've been studying forever. If I'm going over time, let me know. So we've been studying, so let me give you, we never named it nutrigenetics at that time. This is a recent term, but we have known this forever. So phenylalanine is an essential amino acid. I'm sure you all know about this, but it gets converted to tyrosine, and this enzyme, phenylalanine hydroxylase, actually helps in conversion. Tyrosine is a semi-essential amino acid which is needed for us to form dopamine neurotransmitter. But when this SNP is there, either very little phenylalanine is converted to tyrosine, or none. So we don't have tyrosine, and we really need tyrosine. So that's where we nutritionists can jump in. We say, okay, if there's no tyrosine, we'll supplement it in a diet. So it's so important that diet, nutrition and genetics work together. See, if they didn't know what to do, well, what do we do with tyrosine? What types of foods contain tyrosine? Or maybe we give a supplement. And then we also tell them, don't eat phenylalanine containing foods, because it can get deposited in the body, it can be toxic. So this is where nutrition becomes very important. Can anybody think of any other example? It's a very common one I'm going to talk about next, but
caffeine. Caffeine. Any other inborn error of?
No, or an example of nutrigenetics like this.
Folate.
Folate. Folate is one, but folate is more like many genes are involved. This is a classic example.
Homocystinuria.
Yeah, cystinuria. And also, what is commonly when you can't tolerate milk?
Lactose.
Exactly. That's a classic example of, it's actually called lactase persistence. So most of us lose the ability, lactase, which breaks down lactose, we lose the ability to metabolize milk. But we can eat curd or yogurt, but not milk. But those who have this mutation actually have lactase persistence. So you remember that our nutrition habits, your genetics adapt to the nutrition habits, and it works the other way too. So this mainly you'll see that most of the people where milk is a prevalent one, this mutation is very high frequency. Like those Scandinavian countries and all, it's like 90% frequency. And places where they don't consume milk at all, it's like 10%, less than 10% frequency of this mutation. So our body adapts to what we have. So all these small changes in the lactase gene are like how much it is expressed. So all of us have little bit expression, but people who have grown on milk, or they are like dairy communities, they actually can metabolize lactase. Other cases, classic cases, G6PD deficiency. Glucose 6-phosphate dehydrogenase is an important enzyme in our energy metabolism. So what happens is, if you have this, then normally you're fine. You can live normally, but when you eat fava beans, it can be fatal, because the fava beans contain certain chemicals like I don't know how you pronounce it, VRCINE, which interacts with our genetic variants, and it can cause hemolytic yeah. And most of the sub-Saharan Africa and all, they have very high prevalence of, even Parsi Zoroastrians, the Persian area, they all have this, they all get tested for this. And if they know that they have this mutation, then they try to avoid fava bean consumption. So, and then, but none of our like obesity, diabetes are all handled by a single gene, right? There are multiple genes. What we do in that case? Then in that case, we folate, someone was talking about folate. This is where it's a classic case. So we have methylene tetrahydrofolate reductase, which converts homocysteine to methionine. And methionine goes on and helps in methylation and all. But what happens is that if there is a mutation, that activity reduces. So homocysteine is no longer getting converted to methionine, which affects not only folate metabolism, but increases homocysteine concentrations because it's not getting converted. Increased homocysteine can increase blood pressure and can lead to heart problems. So that's why then what we do is folate is not metabolized. Again, we nutritionists step in, we say, okay, if you have this mutation, you should be taking 60 grams more folate than other people, you know. So knowing I'm just giving an arbitrary number of 60 grams, but I'm just saying that we can advise them to eat more folate rather than, so that we can make up the loss of folate work. The other classic case is, I'm sure most of us have this problem, is caffeine. We have some people who say that, oh, I drink coffee even at 10 o'clock and I can fall asleep at 11. But some people say, oh, I don't want to drink coffee after 3 p.m. I won't be. So those who are saying that, they have this mutation in CYP1A2 gene, which makes them slow metabolizers. That means they're slowly breaking down coffee in the system, and coffee stays in the system for a long time. So it won't let you speak. And those who are fast metabolizers, they drink coffee and within a few minutes it's gone from the system, metabolized. And so this is also a classic case of nutrigenomics. Okay? Then what is nutrigenomics? Like I just mentioned, how food affects how our DNA is regulated. And the sequence is not much changed, but how the RNA is expressed and how the protein and finally the metabolite, which is the final product of metabolism, how are they affected by what we eat. And again, folate is a good classic case of this, and many other nutrients can affect. And finally, it affects how health is affected. Like folate, actually folate is a classic case where folate metabolism is affected. But this can also lead to other problems, is people think that they need a lot of folate and they start eating too much folate. So then, now we know studies show that too much folate can cause cancer, certain types of cancer. So we have to stay within the region. All women who plan to get pregnant and all, they should start folate supplementation, but be careful how much folate they are taking. Similarly, recently I was reading a book, reading a paper in which they said, beta carotene is good for eyes. We just tell everybody. But beta carotene in cigarette smokers is really bad. It causes cancer. So we think that, beta carotene is called, blindly we take it, but not for everybody. Either they stop cigarette smoking or they don't take beta carotene. Okay? So it is like combination of these things. Quickly on what is epigenetics, I don't know how many people know about it. So earlier you saw that the nucleotide was changing. In epigenetics, DNA sequence is not touched. Though no nucleotide or A, B, C, D are not changed. But what happens is a methyl group gets added. Many people look biochemistry, they'll understand it. You all have that, right? Yeah, methylation group. So methyl group or acetyl group or phosphorylation, anything can happen. Why DNA methylation? Because it's the most studied. But we know that any other group can also get added. So what happens is, if you have this DNA, methyl group attached to the DNA, how it is expressed, everything changes. So DNA, A, B, C, D are there. And most of the, what nucleotide C gets, especially when C and G are next to each other, that's a very hot spot for DNA methylation. So if you have in your sequence G, C, G, it can be good too. DNA methylation is not always bad. It can be good also. And then histone modification. Histone modification is how the DNA is encapsulated or covered. These are the covered, which can also be acylated or phosphorylated or methylated. And then finally, there are small RNA floating around in the body. So what happens is 22 nucleotides or something. what they do is they go ahead and bind to this nucleotide and interfere with the expression. Okay? So that is epigenetics. It's another area of precision nutrition which can affect that. Then I am sure you've heard of microbiome. Our body is made of community of microorganisms, and it's not just gut. We have many, like eye microbiome. We have many other types of skin microbiome we have. So we have a lot of focus on gut microbiome because they help in digestion, and very interestingly, they also produce their own metabolites, and those can help to. Most of the studies have been done on immune, gut, and brain health, because the brain-gut axis. And then what it does is, some ways bacteria can affect what you eat and how it actually affects the DNA. So if you eat something, it will be like a intermediate point where it will affect how it is metabolized, and finally it has an effect on expression of genes or metabolites. So there are some other omics which are more of an approaches, study of metabolites, like the final product of metabolism, proteomics, how the proteins are, because metabolomics is not proteomics. Metabolomics will give you information on amino acids, fatty acids, but not proteins and lipids. If you're not interested in metabolites, then you should not be doing metabolomics, you should be doing proteomics if you want to understand the protein. Of course, if you want to understand one or two proteins, you will do ELISAs in the lab. And the exposome is a comparatively newer, but we have studied diet all the time. So diet is one of the factors, and it is exposures. It can be any type of external exposures, lifestyle, environment, diet. So just imagine you walk through, you're eating something, okay, and then first gut microbiota acts on it. Some of the foods may be digested there, but some of them may continue further. And then here, genetics or our DNA, what is our sequence for it and how we metabolize. And finally, when it goes, methylation or epigenetic effects can happen. And then, of course, lipids and proteomics, not all of them will affect. But this doesn't mean that everything is affecting, but these are some of the factors which can affect how your food is metabolized, and eventually how it affects the disease risk. This can affect the deposition of plaque or maybe not deposition, either way. So, how do we practically implement? Those who attended the UC, they must have seen this picture, the same one. How I really like this picture. It's mainly focused on diabetes. So if we had data on all our demographics, age, sex, and everything, and then we put in data from microbiome, genetics, and epigenetics, and with metabolomics, and we come up with algorithms, and then we can come up with groups of people who can, and tell them that, oh, you guys, you responded well to this, so you should eat this, or you should not eat this. So at this stage, we are there. Once we have group, then we can go to the individual level. Okay? So how does it apply to food security? I did quite a bit of search on it, and they're not many studies. So I also read that it's a big concern in India. You know, in the Food Global Index or Global Food Security Index, it is 68 out of 130, which is not a really great thing. But so what is it actually? What do you mean food insecurity? So, or food security? So it's basically, is a person getting an adequate diet? Adequate doesn't mean only calories, but proper diet, enough proteins, carbohydrates, and fats, clean water, sanitation, and overall health, nutritional health. So, and how do we check? We check the availability, how far. So we have some studies in the US where we look at how far is the park for them to do physical activity, and how far is the nearest store. If we can't, like the person is too far, maybe the person will say, oh, I don't have time to walk down there. That also is a concern, you know, we don't, so we'll say that, oh, the store is so far from my home, so while coming from office, I can pick up something, or I'll just eat whatever is at home. So those type of things. And a lot of people don't have the access. And then utilization. What is the best way? So like I told you that we did a study when I was doing, So we did in the low income women, and they all ended up, we did a personalized, at that time I didn't, generally nutritional management. And they all ended up, most of them ended up gaining weight. So we were like, that was a real eye-opener for me. I was in my first year of PhD. And so those women said that, you know what, I work three jobs a day. Where do I have time to prepare these recipes you are giving us? I don't have time. I go to McDonald's, which is cheap, $1. Fresh fruits and vegetables are so expensive. So I just buy that and I pick it up on my car and I just go home and eat. Where is the time? So as nutritionists, I think we should also think about whatever we are telling blindly, is it practical? Is it actually, can we do it? Okay? So sustainability becomes very important. Maybe they'll follow 2 or 3 days when we tell them. After that, they go back to their routine. And other thing, we have to remember that it's not easy to change dietary habits. Even if I'm so used to Indian traditional food, even after living in US so long, I eat pizza 1 or 2 days, I really long for Indian food. The 3rd day, I'm like, I need to eat my food. So it's not easy. So these are the key diseases which have been associated with that. Overweight. It will be surprising that we think that food access is so difficult because they end up eating wrong types of foods. And then cognitive, neurological disorders were quite a bit actually, and chronic lung disease and joint muscular. So where can precision nutrition help? So what I found the papers, I'll talk a little bit about, and then I'll tell you what I think. So most of them were based on agriculture. How can we improve crops? How can we do some, it's not really genetically modified crops, but how can we improve to get more availability? Can we save the seeds? So it said that seed banks, we can create seed banks and just store. So this is basically all about agriculture. And how can we modify the agricultural genomics to improve current crops? But this is not really like precision nutrition, but I found a very interesting study on food insecurity and gut microbiome. So I want to talk a little bit about this. This was published in Scientific Reports. A really recent paper, 23. Okay? So what they did when they went to a college and this is the study design which they had. So they actually calculated food security index. And they based on the food secure versus food insecure. So they had you see that N is equal to 38 and 30. And then they gave them a certain diet and they followed them and collected stool samples and then analyzed those stool samples. And this was like a table where they had so many, this is the age group because college students. And it's very interesting, this paper, the authors who wrote this paper, actually their focus was eating disorders. So they were trying to do that, but out of that, this paper was focused on food security and food insecurity. Eating disorders can also be included in food security. So, and then they had different types of ethnicity, a small group for ethnic background, but they had so many age groups and what they ate and dietary. So if you look at the alpha diversity. So microbiome is, it's not a very simple thing because we have so many bacterial species. And right now we don't know, we do have some bacterial species associated with beneficial effects, beneficial health outcomes and some, but it's all very preliminary studies and we're doing more. And so if you see clearly, we have food secure and insecure, there's a clear demarcation, and so with certain food insecure people have high levels of certain species. And then you see that this also, they're clearly demarcated. In this one actually it is confusing, but these species are all clearly demarcated. But we are at a stage where we are doing just associations. Correlation is not possible. We really don't know whether, we're just saying, okay, increased quantity of this species is associated with food insecurity, and these quantities associated with this. It has to be done further work and all. But interesting, isn't it, that I did a lot of search and found one study on gut microbiome and food security. So And then taxonomy basically is each species being named, and you'll see that each of them, but the clearly demarcation, you see that all food insecure are collected here and for all food secure. That means something is happening in the bacteria, or some people who are food secure tend to have certain higher quantities of certain bacterial species. But this is a very good study to generate hypothesis. I won't say this is the final one, but it generates a lot of hypotheses. What's happening in this species? I wish they had, but this paper was on that. I wish they have genetic profile or something. We can go deeper into look at that. What is this doing? What is this doing? But not many are there. So before, I'll just take summary and then I'll tell you. So what precision nutrition approaches are that we can help understand variation in how people respond to nutrients and how these factors eventually affect obesity or diabetes or any other chronic disease. Single gene diseases, monogenic diseases are difficult to find, but once you find them, it's easier to find a treatment. Once you know what gene is affecting this disease, but diseases like obesity, diabetes, multiple genes, and one gene we find, the other will compensate for it. So it's so difficult. That is why it's so difficult to find a cure. And the idea of why are we studying precision nutrition approaches? Why do we care? Because ultimately we want to come up with optimal dietary plans. So the initiative started as precision medicine, where people looked at how people responded to diet, to drugs, pharmaceutical. And then they started looking at some pharmaceutical drugs interact with nutrition. So like, what about nutrition? So precision nutrition came up with that. And I think personally that utilization of precision, we don't have to be very sophisticated, but simple steps. Simple steps we can use to reduce hunger and provide input. Maybe we can come up with less costly food population across the globe. Or if we know that a certain group is susceptible to certain disease or certain problem, maybe we can provide more access. If we know clearly what is happening, if we provide more access. So if we have some time, I want to open it actually for a few minutes and ask, we have 5 minutes, right? So I would like to, I'm very interested to know, what do you think? I hope you have an understanding of precision nutrition and food security. So anybody, any ideas how we can implement? Because there are hardly any studies, so I want to know your views. Personally, I think, yes, we can study how food insecurity affects protein levels, and some people, like 2 or 3 people are food insecure. You give them the same diet, they all may respond. For one, that may be enough, and they become better already. But the other 2, maybe not enough. Maybe we need more to. So that is my idea, that way precision nutrition will help, that we don't blindly give everybody the same, like food insecure, they found 32 people or something, we all give them same diet, they'll all respond differently. So anybody want to talk? Yeah, go ahead.
Ma'am, can we think about, when you're talking about food insecurity, can we think about biofortification and its role with food insecurity and genetics?
Yeah, we can, but biofortification also, we're doing it general, right?
Yeah.
So we may have to add something for people who are genetically susceptible. Yeah, it's a good idea. Biofortification is a very good idea. But you have to do more than plus something for more genetically susceptible groups.
So what we can do is, we can consider one kind of population of a certain disease. Like for example, if you're thinking of biofortification in the form of vitamin A, so we can correlate it with some of the other disease, and we can have a subgroup of the population, and then we can proceed with biofortification there.
That's a very good idea. Yeah. Because in our study, of course, we're not doing food security, but we did group them like that, how they respond to a drink or something. Yeah, that's great. Anyone else? It's a free thing, don't worry about ideas come out like this. Or we can talk about it later. I'll just stop here. I want to thank my team. They make me look good. Thank you. Any questions? We have few minutes, 2-3 minutes. Or you can ask me if you want to.
forum is open for question and answer session. I'm inviting some questions from the students. It's a topic which we have just I think introduced even in our syllabus. But earlier we never used to talk about precision nutrition and food insecurity or security. So yes.
Ma'am, I have a question. While we were talking about the precision nutrition plate, you mentioned that the exposure is not mentioned in the plate. So according to you, can we mention exposure in the precision nutrition plate? Should it be a part of it?
Yeah, I think so. It should be part of it because like exposure can just be like not only exposure to pollutants or something. Any lifestyle, right?
Okay.
How you do physical activity. It also affects how our nutrients are metabolized. So yeah, I think we should include exposures. And there is this paper is slightly old, but there is increasing recognition that we have to include the exposures and not only physical activity. Some people include psychological with more of a biological, but psychological also include. So yeah.
And now we are talking as precision nutrition to be a population subgroup. So how much time do you feel or how many years is it going to take for us to move to the individual approach in the precision nutrition?
It doesn't have, you don't have to wait till precision nutrition is done to this. If we are doing in our search and we find a very interesting subgroup which responds like this, we can move to personalized nutrition. I just wanted to specify that they both are not the same. Oh, that's all. But yeah, we don't have to wait that precision nutrition time is done and then we move to personalized. No, we can do them parallel.
At the time they can correlate, right?
Yeah, they can. Depending on how confident you are about your results, in this population subgroup is really susceptible to something, or then you can start going individually to them. Yeah.
Yes.
I request Dr. Rekha Patalwar to please felicitate.
No. Any more questions? Yeah. Let us. We have time. 5 minutes more. Yeah.
So ma'am, precision nutrition, when it comes to that, it's all genomics and people are not much aware about it, especially because it is also very costly. So shouldn't we first develop some cost-effective strategies and then go ahead with population-based?
That's why I love talking to younger people. They come up with such good idea. Very practical and good idea. Yeah, I totally agree. So I think you guys can come up with new ideas. But when I started my PhD, years ago, but whatever, each whole genome sequence was like $10,000 per person. Now it is 1,000. Even it's going further down. So I am sure we'll come up with. Now we have like this much small box where we can sequence the gene. Earlier we needed big equipment. So yeah, I agree totally, because if I go, the one other point I didn't raise is it's the scare about genetics. If I go to someone and say, oh, I'll give you a genomic sequence, they'll get scared. So how do we review that? So yeah, cost and faith, or people, you have to educate people that genetics is always not like scary or anything. Yeah? So yeah, cost effective. I'm thinking that we will come down. In fact, India is cheaper than a lot of these than US, because you're calculating in rupees and dollars. So yeah, so I'm sure it will come. But I agree that right now it is sort of it's still not like a personal level. We can't just go and if we do it, we'll do like 300,000 markers or SNPs or something. Genome will have millions of steps. So it takes some time, but totally difficult. It's really expensive.
Yeah, thank you so much, ma'am. I think already I can see there are a lot of questions coming up, so we have piqued the interest for all of us here. There is always a gap between the research and the practice. Now, because we are not aware, because we are not qualified, because we are not experienced, and maybe because there's not enough evidence in the literature, so bringing it to a practice level, what are the challenges that you see, and how can we bridge that gap between research and practice? Because unless the dietetic fraternity practices it, it will not reach the population or the public. So how do we go about that, ma'am?
Yeah, it that certain level we have been practicing. There are several companies come and they based on your certain genetic profile, not the whole body, but they'll say if you have these 19 genetic variants, you need this time, which I don't agree with them. Because based on just 19 SNPs, you can't give a diet. You have to know the whole profile. So the dietitians also need a genetic, some type of training or something. So I do summer workshops every year where all the dietitians come in and I train them in genetics. But it's only like a week. So what will they learn? A lot of people follow up with me, and so yeah, they need a lot of training, and we need genetic counselors there so that we talk science, and they do the like parts of people who come in, they get scared. How do we talk to them? So dietitians, we have to train dietitians in that also, the wellness. So some of the like folate is so easy, like if they have this mutation, we can tell them take more folate or something. So you don't have to explain to them that they had this mutation, you just said that you need more folate or something like that. Or some other cases, so we can do that. But we are at not a stage where we are doing genomes, whole genome and testing that. But I think training on both parts, nutrition, people understanding about genetics, and also genetic counseling is becoming very important. They are the ones who need to understand how this diet comes. So it has to be like a team of people, not one person doing it. A dietitian plus a geneticist or a genetic counselor together, how you explain it, and what type of diet you eat. So these things are all kind of done in one person. So I'm part of a big consortium where we are trying to see how to return results to the participants because we feel like they deserve the results. So generally we don't return that, but in this study we are trying to do that. So we are trying to devise the best ways to tell them that based on the results. So that involves registered dietitians, we who do the back work and all together.
I think I totally agree because, and I'm taking the liberty because we have 10 more minutes so we can have more questions.
Okay, sure.
So I will invite everybody else also. If there are more questions, please feel free to ask ma'am. I just wanted to again add on to this here that we have some nutrigenomic specialists or experts who are practicing in an Indian context, so they're very few and limited across special areas, and they also work as a part of a multidisciplinary team. But there's a lot of fear among genetics is like mathematics and a lot of times, and somehow because we learn biological sciences, mathematics, informatics, AI, and now the different omics technology there. So would you suggest that a capacity building simultaneously along with research backing and maybe some industry collaboration, who are the people who are actually preparing these kits and analyzing the reports and sending us the interpretations?
Yeah.
So.
We need a bioinformatics person or a data analysis who can make us. But most of the statisticians or bioinformatics, they don't have any, they don't know much about nutrition or genetics. So it is like I said, it's a combination of people. It's a team. So you have to have one biologist or a metabolism person, and then geneticist, and then of course nutritionist or a dietitian, and then a bioinformatics who can make sense of it's, the problem is that it's extensive amount of data. Like you are talking about like millions and millions of data points. So how do we integrate all that to make sense of it? So that's where your AI can come in. So we need an AI expert or a bioinformatics expert. But AI also we should not blindly do, because remember that computer will output whatever we put in. If we put in wrong information, we get you get what you put in, right? So we have to be really careful, even in AI, what we are putting. Are we doing the right information? See, everywhere technological approaches are so important. Like we are measuring biomarkers, we have to be sure that it is accurate. Dietary information, we still depend on food frequency questionnaires and 24-hour dietary calls. We don't have an objective approach. So there's so many like little what do you call it? Not clear approaches. And even biomarkers, we have to be sure that so quality control is going properly and we are in proper ranges and all. So then we can surely say that yes, this is So people always talk about this, but approaches are very key. If you make mistakes in approaches, you can't trust them.
What would you suggest our student community? Majority of them are very interested in nutrigenomics with what we have understood with our interactions with them. So what would be our next logical step for them?
I think that don't go with fancy words like nutrigenomics and big word, AI and all. Start with the basics. Like if you understand basics, picking up later ones are not. And very simple way. What you want to know is what diet should I eat based on my genetic, or something like that. Start with simple and slowly add one variable to it. Okay, maybe I have done genetics, maybe I had epigenetic portion or something. So if you start doing everything, you'll get so confused that it will take you anywhere. So just keep it very simple, and there's nothing wrong with not knowing. I tell my team that even if you think it's a silly question, ask me, because if I ask a silly question, everybody will think, oh, she's silly. But 5 minutes later, they forget. But if you don't ask that, you'll remain silly the whole life, right? So ask questions. I insist on asking questions because if no question is silly or anything. If you ask questions, that means you want to clarify. So ask questions, go with the simple approaches, and then don't go with this fancy ways. I know they are not good enough, but just understand what it is, and then I think if you go with simple and slowly add complexity. Because I feel like diet plans and all, I find that very difficult. I used to do it, but now, so it's understanding genetics, and you are coming up with diet, and you are actually in touch with the people. You are actually prescribing their body. So you should be very clear in your principles or basic things. You should be knowing very well. So that was diet. So a lot of people use complicated words and all. I don't use, even when I'm teaching, I use like give examples of very simple one, so that it gets into the brain and it will never go. You had a question?
I had similar question.
Oh, similar.
I need a clarification. Like you said that this study is on subgroups. So in India, are there any studies? Because in India, as again, they have this diversity, like South versus North versus West.
I just don't think that. I didn't
Are
see
there
any.
any?
No, I don't think so, because I did a lot of search in last few days since. I don't think any. Even in US, they're not. Europe is very much ahead of everyone, even ahead of US. They caught on to the precision nutrition much earlier than now. US is behind and other countries. But the problem is that everybody wants to talk about precision nutrition. Most of people don't understand what this is. Maybe that is looks very nice. That's why I always say that don't go with this fancy. People just get AI and what is it? It's like a regression analysis and what you feed into it. AI people may not be. But whatever statistical analysis you do, what data you are putting in, that is what you get results. If you put wrong data, then you get wrong results. So be very clear on your methodological presentation. Be very precise. The precise part comes in that process and study design. If you have a proper study design, then later on you can go back to current study design. So spend as much time you can on study design and what approaches you want, and then you can.
So I think our future nutritionists are having this chance of doing the studies and adding on to our Indian literature based on our subgroups. Any more questions? This side, must. Yeah, if we can get those questions, 1 or 2 questions.
Can you just see if other rooms have any
Do they have any questions?
Room or one room,
Yeah,
if they
yes.
have any questions, then we can ask them to come here.
And if you have questions later on, please to ask. I love interacting with students. Mine is Saroja at unc.edu. Just the first name at unc.edu, and just introduce yourself that I attended the talk, or I'm from here, not even, just to know that. Ask questions. And I'll respond.
wonderful session on precision nutrition. And earlier, as we all thought that it is personalized nutrition counseling, but ma'am has focused and said that no, it is on the population subgroups. And then of course from population subgroups we can go to the, like personalized counseling. She has explained to us the interaction between the gut health, okay, various genetic modifications which are taking place, and ma'am has also linked that to the food security or insecurity. That is again a topic which is coming up now about food security, as to how we can improve the food security so that each and every individual or citizen of India is going to benefit out of it and improve their health and the nutritional status. Saroja ma'am, you have done an excellent job. You have come, I think, as you said, very simple, but you have given the valid explanations. I think all our students where we find nutrigenomics and all that going above our heads, and this fancy name of precision nutrition, you have just explained very clearly that it is nothing but what we already know, but we are just giving some terms. That's the only thing. And as ma'am has said, as the budding nutritionist, you have to always focus on the basics. Start with your basics, and then of course you will specialize, and with the experience you come up to that individualized, personalized nutrition counseling based on the precision nutrition. Thanks a lot, ma'am. You have come all the way from US, and for sharing your time with our students, and of course we have also gained out of your session. So thanks a lot for your time with us. And can I request our Vice Principal, Mrs. Manjari Balerao, to give a token of appreciation and love to you from our institution.